review of Chapter 20. Chapter 20 is on DNA technology. Generally, my lecture notes kind of skip around. I pick and choose some of the subject matter and put it together in a particular way. Now, I do start at the beginning of Chapter 20 in Figure 20.2 as an overview of DNA technology. So this figure depicts inserting a gene into a bacterium to make it do what you want it to do. So basically, you have to isolate this gene of interest that you're after. Okay, that's step number one. And then you cut that gene out and insert it into a bacterial plasmid. Now, a plasmid is a little circular piece of bacterial DNA. Then step number two in Figure 20.2 is that plasmid is now put into a bacterial cell. Then step number three, as the bacteria divides, so does the plasmid in terms of DNA replication, making copies of your gene. That's known as DNA cloning. And of course, as the bacterium transcribes and translates that gene, then the gene product can be used for various purposes. For example, human insulin. The human insulin gene can be placed into a bacterium and it will transcribe and translate human insulin just as good as yours and mine. That's how it's mass-produced pharmaceutically. Now, in B of our notes under Roman numeral 1, I talk about restriction enzymes. Restriction enzymes are used to cut and paste DNA. So a restriction enzyme is one that locates a particular sequence on the DNA molecule, makes a cut only at that sequence and nowhere else. For example, here we have a DNA sequence right here. A particular restriction enzyme may make a cut there, but it will cut it unevenly. It will cut it like this, exposing these so-called sticky ends. Now, another piece of DNA containing the gene that you're after can be cut using the same enzyme, having the same sticky ends, and it will fit in here. So restriction enzymes are a way that DNA is cut and pasted. Under C, it talks about polymerase chain reaction. Polymerase chain reaction is shown on Figure 20.8. It is an artificial way to make many copies of a DNA sample. So basically, it uses DNA polymerase, which is what cells use to replicate DNA. This time it's done in the laboratory. So polymerase chain reaction can take a tiny DNA sample and amplify it so that you have enough DNA to analyze. So that's cloning DNA also. Roman numeral 2 of our notes talks about analysis of cloned DNA. You have a picture on Figure 20.9 of gel electrophoresis. Now, what you can do with gel electrophoresis is you can separate molecules from each other out of a mixture of molecules, primarily based on their size and somewhat on their electrical charge. Now, you have a gel about the size of a deck of cards, but only a few millimeters thick, literally made out of a gel, a jello-like substance. You'll have these little holes in there called wells where you load your samples. Then you lay it down on an electrophoresis tray, again, Figure 20.9. You subject it to an electric field, and the electric field will drag the molecules at various distances, separating them. And then you have to stain the gel so you can see these bands where these molecules represent. Now, in B under Roman numeral 2 of the notes, I have RFLP analysis, restriction fragment length polymorphisms. So it's a way to analyze DNA. It's done in DNA fingerprinting. So if you're trying to match DNA samples, let's say you've got 5 samples of DNA and you don't know whether they match any of them or not. Then what you do is you take your DNA samples, all of them, use a mixture of restriction enzymes to cut them into pieces, restriction fragments. And of course, these DNA samples are all now mixtures of pieces of DNA cut with restriction enzymes. Then you run your electrophoresis gel, and the sizes will separate into their bands. The smallest of the fragments will migrate the fastest. The largest of the fragments will migrate the slowest. So in other words, you separate them out over a couple of hours. And then you can stain and view your bands. So suppose your restriction enzymes look like this. like so. Well, now you can compare bands. Samples 2 and 4 match, meaning they likely came from the same individual. Now, the actual cloning of an individual organism is shown in figure 20.8. It's known as nuclear transplantation. Basically, in principle, what you do is you take an egg cell, remove the DNA, get the DNA from the nucleus of the organism you are cloning, and then put all those chromosomes into that egg cell, stimulate it to develop, insert it into a female to carry at the term, and you got your clone. Now, in principle, it's that simple. Making it work is not that simple, but it has been done in some cases. You've heard of Dolly the sheep. Figure 20.19 is the first clone cat, and things like that. Of course, why clone cats? Well, we don't have a shortage of cats. Roman numeral 5 in our notes talk about practical applications of DNA technology. DNA technology such as this is used to diagnose disease. It can be possible to determine whether someone is a carrier of cystic fibrosis or a carrier of the Huntington's allele simply by matching their DNA with the known DNA of that gene. So it can be used to diagnose disease and identify carriers. Theoretically, human gene therapy. A normal gene can be inserted into someone's bone marrow through a bacterial vector and cure any such things as hemophilia, where the person is lacking that particular functional gene. Pharmaceutical products, I mentioned human insulin. Certain vaccines are produced by genetic engineering. Human growth hormone, produced by genetic engineering to treat pituitary dwarfism. Tissue plasminogen activator, also a human gene used to dissolve blood clots in emergency situations, mass-produced by genetically engineered bacteria. So there's all kinds of things. Forensics. Blood samples can be matched from violent crimes. Environmental uses. Engineered bacteria can eat oil spills. Agricultural uses. Crops and livestock can be genetically engineered to grow faster or be more pest resistant or be more nutritious. Golden rice is one example. Golden rice is given a gene to produce beta-carotene in the rice kernels, which is useful in countries that suffer from vitamin A deficiency. They can eat the golden rice and get their vitamin A. So there's all kinds of situations. Now in Roman numeral 5, I mentioned genomes. I actually pulled this information from chapter 21. That's about the only thing I'm pulling from 21 for us. This is table 21.1. The only thing I wanted you to pay attention to is genome sizes. Now we have on this table genome size in terms of how long the DNA is, the number of gene loci the organism has, and the average genes per megabase pairs. Now humans have about 20,500 gene loci, okay, something to that effect. The average eukaryote has somewhere between 20 and 40,000 gene loci. Prokaryotes will have far fewer. And that concludes our notes from chapter 20.